Welcome. My name is Charles Sini, founder and managing director of New Tech at the Nanopoint. The following video, titled Functional Performance Parameters of Magnetic Nanovectors based on Superparamagnetic Iron Oxide Nanoparticles, discusses various aspects of the behaviors of magnetically responsive nanoparticles, from synthesis and characterization to clinical application and commercial utility. As the therapeutic focus shifts to individualized or personalized medicine, technology platforms are being sought that can cause or enable a desired physiological event to occur primarily at a target site without harm to healthy tissue and organs. This is especially true in cancer chemotherapy, where cytotoxic drugs often have unwanted side effects that affect both dosing limits and patient compliance. The application of SPION magnetic nanovector technology to the miniaturization of biomedical devices and therapies is an emerging field, anticipated over the coming years to deliver a range of new therapeutic tools that will offer the patient and physician new and more effective treatment options. This paper presents a discussion of SPION performance parameters in terms of their projected impact on emerging clinical procedures, when SPION constructs are derivatized either for delivering an action at a target site or as a vehicle for carrying a biologically active agent to a target site. Their behavior following systemic administration can be influenced with the application of an external magnetic field gradient. Superparamagnetism is a fundamental property displayed by single-domain ferromagnetic materials that occurs as a result of a reduction in grain size. At a grain size of approximately 40 nanometers in diameter, single-domain nanostructures will dominate, and certain intrinsic properties are altered, resulting in the SPION product. In this state, the magnetic susceptibility increases by orders of magnitude, and the nanoparticles now have the capacity to undergo spontaneous magnetization and demagnetization, leading to the concept of a biomagnetic switch. This property can enable the development of a variety of clinical applications in the emerging field of nanomedicine. Functionalized SPION will be rapidly magnetized in an applied external magnetic field, followed by rapid demagnetization without remnants or residual magneticity when the field is removed, as demonstrated in the figure which shows the hysteresis loop of single-domain SPION. The origins of the magnetic nanovector technology platform, as shown in the table, began in the late 70s with the work of first Witter, then followed by Luby, who explored the feasibility of magnetic targeting, where some initial but limited success was achieved. The history extends to today with the work of Sini and Klostergaard, who in 2012 demonstrated tumor reduction in a small animal xenograft model. For the work reported here, SPION synthesis has followed the Massard sol gel procedure, with the resulting black gelatinous precipitate being redispersed using citrate complexes and hyperdispersants to create stable nanodispersions. Silica treatments followed the Correa-Duarte procedure, with silane linker chemistries then employed to attach terminal functionality, through which subsequent biofunctionalization was achieved. Characterization of particle size and particle size distribution was accomplished through dynamic light scattering. Composition and crystallinity was accomplished through X-ray diffraction. Magnetic susceptibility and remnants was measured through vibrating sample magnetometry, and the effectiveness of silica treatment as an inhibitor of the Fenton reaction was measured by electron spin resonance. To be useful in biomedical applications, surface treatment of SPION is required in order to eliminate the capacity for free radical generation, as occurs in the Fenton reaction, which can have negative biological side effects. Silica surface treatment also provides a flexible platform for subsequent biofunctional modification. The effectiveness of silica surface treatment to eliminate SPION capacity for free radical generation is shown in figures 3 and 4. In figure 3, ESR analysis of silica-coated SPION constructs demonstrated no capacity for free radical generation compared to a DMPO control. In figure 4, a dispersion of silica-coated SPION in a solution of sodium thiocyanate over a period of 2 days at ambient temperature, followed by 2 days at 40 degrees C, showed no deterioration versus a control of naked SPION. Although SPION constructs demonstrate numerous properties that can be useful in the development of clinical platforms, it has to be demonstrated that derivatized structures, which add non-magnetic components, continue to meet the requirements for magnetic-based performance properties, and that the key critical characteristics of magnetic susceptibility and single-domain structure are retained. This is demonstrated in figure 5. The top half of the graphic illustrates the changes that occur in the average particle diameter and size distribution as naked SPION, 5A, is first treated with silica, 5B, then complexed or functionalized with paclitaxel, 5C. The bottom half of figure 5 shows the impact of these changes on key magnetic properties of susceptibility and zero remanence. It can be seen here that after functionalization, the SPION constructs retain both magnetic performance and nanoscale identity. Another significant question is whether or not SPION constructs can generate a force at a target site that can be useful in facilitating or supporting a desired physiological event. In figure 6, we demonstrate this using an experimental setup that includes a digital manometer to measure the pressure drop that occurs as a magnetic force is exerted on a silica-coated SPION dispersion at a point opposite the manometer arm. The pressure drop shown by the manometer is due to the magnetic force that occurs in the direction of the tandem magnet sitting opposite the middle arm of the horizontal tube containing the nanoparticle dispersion. That increases with the deposition of magnetic nanoparticles at the target site. This external force being generated at the target site can be useful in overcoming physiological barriers such as the interstitial fluid pressure exhibited by many solid tumors. The IFP represents an obstacle in cancer therapy because cell penetration is resisted, thus reducing the uptake of therapeutic agents. The external magnetic force can also be useful in driving an increased extravasation outcome via the EPR effect to enable more active agent to reach the tumor vasculature. This external physical force will enhance the efficacy and efficiency of magnetic-based therapeutic platforms. Now that we have demonstrated the preparation and characterization of SPION and derivatized SPION constructs, it is necessary to show that these constructs retain key performance properties of magnetic susceptibility over a distance, sufficient to be influenced by externally generated magnetic field gradients. In the video demonstration, the experimental setup includes an array of permanent magnets shown on the left and a magnetometer probe attached to a vial on the right, situated at a distance of 3 inches from the magnetic pole face. The magnetic strength at this distance is recorded by the digital magnetometer shown at the top, which registers approximately 130 Gauss. As the vial containing the SPION constructs is brought into the magnetic field, particle-to-particle interactions can be observed in the vial as the magnetized SPION constructs attract and build structures. Thus, we observe that silica-coated constructs retain magnetic functionality and demonstrate magnetic susceptibility over a distance, a fundamentally important property for magnetic nanovectors. Further, to be useful in clinical applications, it is also essential that SPION constructs respond to the external magnetic field gradient while under high flow conditions, such as would occur when circulating through the bloodstream. Under such dynamic conditions, SPION must continue to demonstrate the properties of magnetic susceptibility over a distance sufficient to be influenced by externally generated magnetic field gradients. This is demonstrated in the video, which utilizes a dual-channel flow-through cell, with the two sides or channels separated by a cellulosic membrane having a nominal pore size of 400 nanometers. A SPION dispersion of 50 mg per mil is pumped from the reservoir below through the back side of the cell at a rate of 150 mils per minute and then back to the reservoir. On the front side, or static side, the tandem magnet combination is brought to a point on the face of the cell, which is a distance of approximately 1/2 inch from the membrane. As is observed over a period of a few minutes, SPION constructs are drawn through the membrane from the nanodispersion circulating to collect at the target site. Successful application in the clinic, in which SPION are caused to move to a target site. An external force is necessary such that SPION behavior can be controlled and manipulated from a distance. In our studies on targeted drug delivery, the magnetic field generator is based on arrays or design configurations of neodymium permanent magnets, which both shape and focus a magnetic field gradient that encompasses the target site, such that when SPION enter the magnetic field, they would be activated and drawn to the target site. A series of photos on the right demonstrate how arrays of small neodymium permanent magnets in design configurations can generate magnetic field gradients with varying shaped focus response surfaces at a distance of a few inches from the pole face. The response distance can obviously be increased by using larger and larger magnetic arrays. An example of how the magnetic nanovector platform can be applied in the clinic is visualized in the graphic below. SPION, after systemic administration, are caused to move and concentrate at a target site under the influence of the external magnetic field generator, an array of permanent magnets which encompass the target site. SPION enter the magnetic field gradient and are magnetically activated and drawn to the target site. Constructs still circulating demagnetize on exiting the magnetic field and circulate until cleared via normal channels without toxicity to healthy tissue. The magnetic nanovector platform can have several clinical applications, such as targeted or localized delivery of therapeutics, especially chemotherapeutics, enhanced delivery of genetic materials as in gene transfection, placement of SPION for subsequent hypothermia therapy, enhanced delivery, and selective placement of stem cells. The current objective for advancing magnetic nanovector technology as a therapeutic platform, especially in molecular oncology, is to extend beyond current preclinical studies to clinical assessments of both superficial and more visceral or deeper-seated tumor sites, then to move beyond cancer therapy to the treatment of other health diseases. In the figures shown are magnetic configurations presented in two-dimensional visualizations that are designed for treating both superficial tumors on the left and for reaching deeper-seated tumor sites on the right. The SPION magnetic nanovector platform is a drug-device combination that uses an externally generated magnetic field gradient to manipulate the behavior of SPION-based therapeutic constructs to drive a desired physiological event, delivering enhanced benefits and effectiveness to both the clinic and the patient. The anticipated benefits in the clinic and to the patient are shown in the respective tables. Listed here are selected references that present research results in areas of 1. SPION synthesis and characterization, 2. SPION bifunctionalization, 3. Magnetic vectoring of SPION constructs, and 4. Efficacy of SPION paclitaxel constructs in small animal xenograft models. This concludes my presentation. I hope it has been received with interest and that it will prove beneficial to ongoing research. My contact information is shown at the bottom of the biographical profile. Please feel free to contact me with your comments and suggestions. I thank you for your attention.